essential for research to campus. Tim was learning about microbiology and about some of the things that we don't typically associate with bacteria. In this case, their ability to produce electricity. Well, it kind of lit a fire under Tim, and the thing about Tim is we have trouble ever getting him to leave the lab. And his efforts, I think, are very much evident in what you're about to hear. Just last week, Tim presented at the American Society of Microbiology Regional Conference and won first prize in the environmental microbiology division. So we're very, very proud of him. And with that said, let me let Tim share with you the work that he's been working on.
Of course, what I'm going to be speaking on today is my development of a test vessel for microbial fuel cells. And microbial fuel cells are basically bioelectrochemical systems that convert chemical energy into electrical energy through the metabolic respiratory activity of microbes. This is not a new concept. It was first touched upon by Professor Potter at the University of Durham in 1911, where he first considered the concept. But not much followed through until 1931, where Barrett Cohan managed to, through the process, get 35 volts, but only 2 milliamps of current power. So it wasn't very much, not much really took place till about the 1970s. And for those who may know that it was during the 1970s, there was a interest in renewable energy, because during that time we had the energy crisis. So from the 70s, things began to pick up in this area. 1980s, late 1980s, significant advancement took place when Dr. Lovely came across a specific species of microbes, Geobacter. And it had the ability, it was called electrogenic, which I'll explain in a moment. During the 90s, the past 10 years, is when significant advancements have been being made. They're actually able to start harnessing some of the power from these two low voltage, low power supply source. So with that discovery in the late 80s and in the past 10 years, and also coupled with the fact that we have green energy pursuit today, this is taking on new significance. Begin with the microbes themselves. There's two different types of how these batteries work. There's the mediator type, where it's chemically assisted. Additive is put in with the microbes to help the electron transfer to complete a conductive circuit. The other one, which is what I focus on, is a mediator-less type, where it is more directly involved with the microbes who have electrochemically, proteins, active sites on their within their membranes that is more directly allows them to discharge directly to an electron receptor. And there's generally, well, if you look right here, is what I'm talking about, is you got, this is a Shillnella type microbe. And if you can see these wires, which may not be too clear for those back there, they're basically, it's a biofilm that forms nanowires. And the community can get together, and that can cause a current to flow as they discharge the electrons that are building up in their cells. The other type is the Geobacter. If you can see the blue filamentous peeler right there, those are specialized organelles or organ structures on the cell that discharge, that is specifically for the discharge of the electrons. And they usually incorporate a mineral of some type, in this case mostly iron, that is in the soil, that has an electron receptor. And this right here is a possible metabolic pathway for the discharge. There's still research in that area that is trying to develop in how exactly this mechanism works. There's several different types of fuel cells that are out there. There's basically a two-chamber where you got the anode and the cathode on one side. The anode is plated with usually a single species of the microbes, allowed to grow, and then over time it begins to develop a current and it completes a circuit. There's another type that is a benthic, where a carbon graphite pad is placed onto the river bottom, and the cathode pad is kind of floated up above. A wire connects them that'll complete the circuit. The other type is a two-chamber with a salt bridge, which basically is the same type. It's an anode and cathode chamber, and a circuit is completed with wire connections, with the microbes generating electricity, the hydrogen going off to the cathode, mixing with oxygen, and completing the cycle. Well, when I first became aware of this with Dr. Kiner, I was wanting to try to develop further some of my understanding of lab work and some of the process of the courses that I've been learning. So he was doing some work with microbial fuel cells there, and I asked him what was taking place, and they needed to get something to do a presentation to get the cells active. And usually what I mean by active is after a certain period of time, from 5 days to 2 weeks, is how long the process usually takes before there's usable, visible energy in the process. We're able to light up a light, or in some cases, a small calculator or small fan. From my courses I just completed a course during that semester with Professor Iyer and Dr. Harless. I found out in cell culture that when you remove a cell out of the body, you may be able to make it function or grow it, but it'll never be at its full potential because it's being separated from inside the body where there's hormones and other things that play. And then from general microbiology, we talked about symbiotic relationships. My idea was it didn't make sense separating species. There should be already an established community there that is working together that should be more potentially efficient in the process. So I asked Dr. Kiner for permission to try a hypothesis. I said, I want to try to take a cell, get an established microbial community, and see what it does. And when I did that, and I put it in the cell, I activated, I had a reaction within 24 hours. So based upon that reaction that's substantially cut down 5 days to 2 weeks to less than 24 hours, I had one more theory. And it had to do with how does nature take advantage of its environment. And of course, the first thing I thought of was trees and the root system. So I decided to, what is normally done with a pad, I decided to design a grid where I would plug that grid into the pad. And what happened was as soon as I hooked it up, there was immediate power and more than double what they were getting before. And the cell that I developed is this first prototype.